Right, thank you. Thanks very much for inviting me to give a talk here today. I'm Dr. Tertia Purves-Tyson, and I lead the preclinical neuropsychiatry laboratory at NeuRA. So, I don't know if you can see the pointer. In the preclinical neuropsychiatry laboratory, we use post-mortem tissue, rodent models, cellular models to try and understand the neurobiology of psychosis. We use animal models because we can model different behaviors that we can relate to things that we see in schizophrenia. For instance, we can look at psychosis-like behavior, and I'll explain a bit about that as I go along. So in the animal models, we can look at different behaviors, we can look at molecular changes in multiple different brain regions, and we can relate these things to one another. So we can really start pulling out different behaviors that relate to different symptom domains and try and determine which molecular pathways lead to those specific symptoms, and to try and inform. And in the animal models, then in a reverse translational manner, I take molecules that have shown promise in the clinic for symptoms of schizophrenia and use them in the animal models to try and alter different behaviors and then try and discover the underlying molecular changes. And in the animal models, we can also look at different changes in blood molecules that we can then also try and relate to changes in the blood of people with schizophrenia. So we know that dopamine dysregulation is core to psychosis. We know that too much dopamine in the striatum underlies psychosis, and we know that the cell bodies of the dopaminergic neurons that dump too much dopamine into the striatum are found in a part of the brain called the midbrain, which has two subregions, the substantia nigra and the ventral tegmental area. And these are the regions of the brain that I'm really interested in understanding the molecular changes that might contribute to symptoms. So we know there's lots of different contributors to the pathophysiology of schizophrenia, and this is actually one of the reasons that underlies the difficulty in treating schizophrenia, because it's maybe could be referred to as the schizophrenias. There's lots of pathways into the same set of symptoms, which is why we need to understand the neurobiology of the different symptom domains. So we have the neurodevelopmental hypothesis, which suggests that something goes awry during the development of the fetal brain that predisposes a person to psychosis later on in life. And that predisposition might be elicited by the person's environment. We also have the neuroinflammatory hypothesis of schizophrenia, and Cindy's talk was alluding to that, where we've discovered that there is evidence of neuroinflammation in the midbrain, in the substantia nigra and the ventral tegmental area, that may contribute to the dopamine dysregulation. And microglia are the main immune cells that reside in the brain, and we've discovered evidence that microglia might be altered in the midbrain in people with schizophrenia. And then there's also the estrogen protection hypothesis of schizophrenia. So we know that men might be diagnosed at a higher rate and might have a worse course of the illness than women. Importantly, in women, psychotic symptoms fluctuate with the menstrual cycle, and psychosis is ameliorated at times of high estrogen. And we know from clinical trials that estrogen can improve psychotic symptoms as well as negative symptoms and cognitive symptoms. But estrogen obviously is not so great to be used in men. It has feminizing effects. So people have tested selective estrogen receptor modulators in the clinic, one in particular called raloxifene, which doesn't have these feminizing effects. And they've shown that adjunctive raloxifene actually reduces psychosis in schizophrenia. And this is a meta-analysis of 9 different clinical trials. And we can see that psychotic symptoms have been reduced. But in the 560-odd patients who were involved in these 9 different clinical trials, there was a preponderance of premenopausal women, and only a couple of the trials included men and younger men and women. So there's much still to be learned about how raloxifene might improve psychosis in different sexes and at different ages. Now, I'm showing this slide because it shows an important sex difference, although it's focused on cognition. And what we can see here is that, I don't know if you can see my pointer, that females in blue circles that were given raloxifene had an improved cognitive score compared to females that were given placebo down at the bottom here. And conversely, men that were given raloxifene down there in the blue square compared to the red square over there had a decrease in cognitive measures. And this highlights a really important sex difference, and that we really need to understand sex as a biological variable in all clinical trials and all studies. So in a reverse translational manner, I've taken raloxifene back to the animal model to ask, how does raloxifene work, and how does it work in males and females? And I can look at different behavioral correlates, and I can look at molecular and cellular mechanisms in the brains of the rodent model. And why do we want to do this with raloxifene? Well, there's two aspects. We want to understand how it works to alter different behavioral profiles, because then we can understand the molecular mechanisms downstream of raloxifene targeting the estrogen receptor, and we might be able to identify novel molecular targets for specific symptom domains. And the other thing is we would like to sort of inform more clinical questions using raloxifene. Who can we treat, and which symptom domain are we treating? Are we treating men, women, psychosis, cognition? So the animal model that I use is the rodent maternal immune activation model of dopamine dysregulation. And this model's been developed because it's known that infection during pregnancy is a small risk factor to develop neuropsychiatric symptoms later on in life. So how this model works is that pregnant dams are given a viral mimic when they're pregnant, and the offspring of these dams show behaviors that we can relate to things that we see in schizophrenia. So particularly for this talk, the offspring show sensitivity to amphetamine because they have more dopamine in their brains, and if you so they're sensitive to amphetamine. Which is similar to people with psychosis are sensitive to amphetamine. And there's other things we can measure in these animals as well. This is a very complicated schematic, but essentially we have a dam getting a viral injection or saline injection, then we have the offspring are born, and when they reach, in this slide, when they reach adulthood, they're given raloxifene daily for a period of time, a couple of weeks. And in that couple of weeks, it's shown by the green arrow there, we can measure different behaviors. But for the sake of this talk, I'm only going to talk about the amphetamine-induced locomotion. So if the more sensitive the animal, the more dopamine the animal has in the brain, the more they're going to run around when they see amphetamine. At the end of the study, we take the brains, so we can look at multiple brain regions. We take blood, so we can relate molecular, and in the brain regions, we look at different, we can look at transcriptomics, proteomics, metabolomics, and so on. We take multiple brain regions, that's that whole list there, but as I'm focusing on the substantia nigra and the ventral tegmental area. And of course, importantly, in the rodent models, we're looking at both sexes. I'll skip over that slide. So in this slide, this is raloxifene in male MIA offspring reduces MIA sensitivity. Now, there's one take-home message from the slide. If you look at the top line, on this axis, we've got the distance moved, and then the bottom, we've got time bins. You can see where the amphetamine injection is given. That dumps dopamine into the brain, and the animals run around, and we can measure how much they run around. The more they run around, the more sensitive they are to amphetamine. So the top bar is actually what I'll refer to as a healthy animal. There's no maternal immune activation, and they're increasing their amphetamine sensitivity when they're given raloxifene. But importantly, the animals that have had MIA offspring, they've had this prenatal immune insult, they're actually, when they receive raloxifene, they're reducing the amount of time they run around. So you can see that on the bar graph, you can see the difference in the vehicle raloxifene compared to the MIA raloxifene. And that tells us that the MIA animals, something in their brain has changed that makes them raloxifene reduce their sensitivity to amphetamine. And the important question there is, if that indicates a certain pathophysiology, how do we identify those people that are going to respond like the blue line to raloxifene versus the pink line to raloxifene? So my job is to go and now look at the brains and see if we can determine what the underlying neurobiology of that is. So I've already said that neuroinflammation may be involved. This is a very busy slide, but what it's showing is 6 different microglial-related transcripts that are measured in the substantia nigra. But there's 3 take-home points here. First of all, some of these transcripts at baseline show sex differences in their expression. This is really important. It tells us that whether it's a male or a female rodent, they may respond differently to inflammatory insults because their microglial profile is different. Then the next point is that some of these molecules are changing in response to maternal immune activation. So maternal immune activation is altering the microglia. And then some of the transcripts are changing in response to raloxifene. So both raloxifene and MIA, both MIA and raloxifene are impacting the microglia in this brain region, which is critical to the amphetamine-induced locomotion that I was showing. That's a bit of a funny picture. Right, so this is the last, second last slide. This data is actually hot off the press. You can't see the graph so well, but essentially what this shows is microglial soma size. Larger soma size is generally thought to indicate increased, more active pro-inflammatory microglia. And you can see, if you look, this pointer is not working very well. If you see in the MIA animals, you can see that the soma sizes of the microglia are actually larger than the soma size in the saline-treated animals, in both males on the right compared to and in females. So just to conclude, that what this data shows or suggests is that both MIA and raloxifene may impact neuroinflammation to alter dopamine dysregulation in the substantia nigra and the VTA by altering microglia. So the questions I'm asking now are microglia and their related inflammatory pathways potential treatment targets to improve psychosis? And if that is the case, would this be in a sex-specific manner? Would there be a different treatment for males versus females, for instance? And then I also want to now go back and look. I've got the blood from these animals. We want to look and see if there's any peripheral markers that relate to these biological changes. And then questions we would like to answer in the future is if we identify peripheral changes in the model that relate to the behavior and the biology, can we then take these to the clinic and can we help inform clinical questions as to who may respond to particular neuroinflammatory treatments? Thank you.